Hey everyone, welcome back. In our last video, we wrote a Python script to read a single DNA sequence from a FASTA file. But what if your file doesn't have just one sequence? What if it has 10 or 1,000? So today we are tackling exactly that. We are going to learn about the multi-FASTA format and adapt to our code to parse multiple sequences from a single file. This is a crucial skill for handling things like sets of genes, protein families, or sequencing reads. So yeah, let's dive in. So multi-FASTA file is exactly what it sounds like. It's just a standard FASTA file that contains multiple sequences, one after the other. So each sequence has its own header line starting with a greater than sign like this one, and followed by its sequence data. When the parser sees a new greater than sign, it knows the previous sequence has ended and a new one has begun. So yeah, let's create an example. I'll make a new file and I will name it genes dot faster. Okay, so here I'll create some three short sequences, gene sequences here, as you can see, and yeah, as it's just the three blocks of headers plus sequences. Now, how do we read all three of these into Python without them getting mixed up? I'll save this file. Okay, so if we used our old code in this file, these ones, it would fail because it would override the header with each new one it finds and just mash all the sequences together into one long string. So we need a better approach. So what to do? Right. So the perfect data structure for this job is a Python dictionary. We can use the sequence header as the key, like this one. I'll close this file. So like what we will do is we can use the sequence header as the key and the sequence string as the value. This way we can store and retrieve each sequence by its name. Okay, so yeah, let's write the code. The logic is a bit different, so pay close attention, right? So sequences is equal to blank, right? Then we will do this current sequence ID is equal to none to keep track of the current sequence. Then with open, we will open our file change.faster, right? And in read mode as f. Okay, I think we are putting this in a wrong cell, right? Yeah, we'll put this in a code cell, right? So for line in f, okay, we will first remove leading or trailing white spaces using our command that we have been using since last time, line dot strip. Okay, now if line starts with the greater than sign, then we tell it that old sequence has ended and a new one is starting, basically, right? So it's the key for our dictionary. So current sequence ID is equal to line 1. Okay, so stores the ID without the, this one will store the ID without the greater than sign. Now sequences current sequence ID is equal to blank. So we will initiate the sequence string here, right? Then else. This is a sequence line, so we will append it to the current sequence. So if current sequence ID sequences current sequence ID, then add it to the current line, right? So then let's see what we got. So we will add a print line at the bottom, print sequences, right? Now let's walk through it. we start with an empty dictionary called sequences. Then we loop through each line, and if a line starts with a greater than sign, we know it's a new sequence. We grab the header text, and I'm using this command line 1 to rest to slice off the greater than symbol itself and use it to create a new entry in our dictionary with an empty string as the value. We also store this ID in current sequence ID. Okay. If the line is not a header, it must be sequence data. We simply append this line into the string value of the current sequence ID we are working on. Okay, when we run this and print the result, look at that. A clean Python dictionary with each gene ID pointing to its complete DNA sequence. This is perfect, right? As you can see here, gene ID and its sequence, then other gene ID and its sequence, and the third gene ID and its sequence, right? So there we have it. Perfect. Now working with our dictionary of sequence, right? So now that our data is neatly organized, working with it is super easy. We want to know the length of the mouse gene, so let's access it by its key. So what we will do is mouse gene ID. I think this is a code cell. Okay, mouse gene ID is equal to gene B mouse gene B mouse P53 tumor suppressor, right? P53 tumor suppressor, right? Then mouse gene sequence is equal to sequences and the mouse gene ID, right? Now let's print the length of the mouse gene is and len for the length mouse gene sequence, and we add base pairs here. All right, so let's check here. We have it 119 base pairs of this sequence, right? Or maybe we want to calculate the GC content for all the genes in the file. We can just loop through our dictionary, because here we are accessing all the genes, right? Not just one gene. So we have to run a loop for this. For sequence ID, sequences in sequences dot items. This is the command we use for accessing items in a dictionary, right? Sequence length is equal to len sequences. Then let's calculate the G count is equal to sequence, sorry, this should be sequence, because we already have a sequence assigned. So this one has to be sequence for sequence and sequences, right? Sequence dot count G, right? And similarly C count sequences dot count C. Now GC content is equal to the same formula we use always, G count plus C count divided by sequence length into 100. Now we can split the ID to get just the gene name for cleaner printing, right? So gene name is equal to sequence ID dot split, and we will split it by a certain distance, one gap and 0. Now let's print it. print gene name is gc content is, and then we print gc content dot 2f and percent, right? This should do it. How cool is that? We have built a robust tool that can load any number of sequences from a file and is ready for any analysis we want to run. Okay, so to recap, today we learned how to parse a multi-FASTA file by using a Python dictionary to store each sequence with its unique header. This is a huge leap as you can now handle the vast majority of sequences file you will encounter in bioinformatics. We have spent a lot of time with DNA. In the next video, we are going to take the next big step in the central dogma of biology, that is translating DNA into protein sequences. It's going to be a fun one. So yeah, don't forget all the code and the sample and FASTA files will be on GitHub. Link will be in the description soon. And if this video helped you, please give it a thumbs up and subscribe so you don't miss the rest of the series. See you next time.